Hello, my name is Katie Daniel. I am a pre-health post-bachelorate student, and I have been working on the cloning of beta-glucosidase and Lactobacillus plantarum with my faculty member, Dr. Andrus. This is just a brief overview of my presentation and what all I will be covering, mainly so you know how close I am to being done if you get bored. Just a brief background on my presentation. With what piqued my interest is that with a increase in popularity of craft breweries, there's also an increased demand for making unique beers. One of the more unique tasting beers, I would say, is a sour beer. Sour beers are made by using lactic acid bacteria, and Lactobacillus plantarum is one of the key bacteria that are used for this sour beer making process. It can be used for this process because they produce multiple enzymes that not only improve nutritional value and vitamins in certain foods, but they also develop aroma, flavor, and textures of these food and beverages. And they do this by producing these enzymes, but one of the key enzymes produced is a beta-glucosidase enzyme. And this enzyme actually releases the aromatic compounds that reside in fruits, which allows for the natural flavoring. So you'll often see sour beers are accompanied with a fruity flavor, such as mango or guava. MALZ, which is the gene that I am looking into for this project, is the gene that codes for the enzyme maltodextrin glucosidase, which is a beta-glucosidase enzyme. To begin with, I grew new cultures of Lactobacillus plantarum by using cultures that were provided by Dr. Andrus of previously grown Lactobacillus plantarum. I grew these at 37 degrees Celsius overnight in anaerobic conditions. And with the success of growth on these cultures, I was then able to isolate genomic DNA using a Promega's gDNA purification kit. After I was able to isolate the genomic DNA, I then made primers to be able to run PCRs using this genomic DNA. I made the primers by identifying the open reading frame using NCBI, and then used a program called Primer 3 Plus to create primers that had a proper annealing temperature while also including my full open reading frame for my gene. I then moved on to the actual PCR reactions. This took multiple tries in which I had to adjust the annealing temperature as well as genomic DNA concentrations, and finally ended up running two different PCR reactions, one in which I used my primers for my gene of interest, and then another in which I used primers for the 16S gene that all bacteria contain. This allowed me to ensure that my genomic DNA that I had previously extracted was still viable, and I wasn't running reactions on DNA that was no longer able to be used. I used gel electrophoresis to analyze all of these results. I ran not only my PCR product, but also my genomic DNA product on 1 to 2% agarose gels that were stained with ethidium bromide, allowing me to visualize bands or not visualize bands if they were unsuccessful. I then proceeded to do my PCR cleanup after I was able to run a successful PCR. This PCR purification was performed using protocols outlined by QIA Quick PCR Purification Kit, and this allows me to have PCR that's been clarified so that I can use it for future ligations into my vector of choice. For my growth of Lactobacillus plantarum, I visualized growth on all three plates. This means that I had colonies that I could then use for genomic DNA extraction. As you can see in the figure, this is what my bacteria looks like when it's grown on a plate. I then moved on to isolating my genomic DNA. I ran this sample on a gel, as I previously mentioned, and I was able to visualize a band, as you can see in the top left column. The visualization of a band indicates the success of the genomic DNA isolation, allowing me to move on to PCR amplification. I then ran these two products that I previously mentioned, both the PCR product using my primers and the PCR product using the 16S primer, alongside of a molecular weight marker for the gel. On the left is the molecular weight marker, and then in the following column, I have the PCR product with my primers for my gene of interest, and then the third column exhibits the PCR product that was my control with the 16S primers. Both products exhibited bands corresponding to the proper marker on the molecular weight marker, allowing me to conclude that the PCR was indeed successful and I could move on to the PCR purification. The PCR purification was also visualized using PCR, allowing me to verify that I did indeed clarify my PCR properly. With proper PCR clarification, I can then move into ligating the PCR product into a vector. Once I've done that, I can then transform the vector into E. coli. Once the vector has been transformed into E. coli, I can then grow the E. coli on a culture that has been treated with ampicillin, known as culture selection. Once this has all been completed, I can eventually utilize the E. coli that now has my vector in it to mass produce my enzyme of choice. For my ligation into the vector of my choice, we are looking into using the HoloTag Flexi vector. This is where our previous made primers also come into play, as I have put on restriction enzymes that allow me to cut my gene and kind of paste it, if you will, into this vector. This vector is unique in that it not only has an ampicillin resistance gene that allows for the culture selection, it also has an additional Barnase gene, which is a kill gene. So once transformed, only the colonies that I grow will contain my transformed vectors, or the vectors that have my gene of choice in them. This is a gene map, or a gene cloning map, exhibiting how I will insert my gene into my vector of choice. For the future, I can then transform the gene into the ligated vector into E. coli. Once I have put it into the E. coli, I can then grow the E. coli, as I said previously, on an ampicillin culture. And after I've been able to select for colonies that have my gene of interest, I can then exploit them to mass produce my desired enzyme. These are the resources that I used, and I would also like to give a special thanks to Dr. Andrus for his cultures and continued guidance at every stage. If you have any questions that you can't think of now that I can't answer for you immediately, I'm more than happy to answer any. I've included my email on this slide. Thank you.